Well, I think we can go ahead and get started. It looks like we're staying pretty consistent on our attendees now, so I assume most folks have joined, although of course people are free to tune in a little later in the process, but we can go ahead and get started right now. Hello everyone, I'm Max Herzog, I'm a program manager with Cleveland Water Alliance. Super excited to have you on today's session. Appreciate you taking your time out of your busy day to learn a little bit more about low-cost nutrient monitoring and citizen science. For those who aren't familiar with us at Cleveland Water Alliance, we're a nonprofit economic development organization based in Northeast Ohio, but working across the Lake Erie region. Our focus is really on new technology and how it can drive water quality and related economic development for the Lake Erie region. So we partner with folks from Detroit to Buffalo, working with academic partners, folks in the private sector, government entities and utilities, really a whole cross-sector group of folks that are interested in this intersection of innovation and water. We've been really excited to be able to work in partnership with the team that we'll be presenting today to dive into some very kind of grassroots focused action that's driven by technical tools, as well as some of our more kind of corporate engagement. So that's us. You know, we have a whole suite of projects. Those of you that know us from the past may know us from our Erie Hack competitions, which we run, focused on open innovation. But we really run the gamut in terms of innovation-related programming around water. Kelly, can we go to the next slide? Is it advanced? Okay, thank you. So this presentation is kind of spinning out of one of our big projects right now, which is focused on, or driven by, I should say, a partnership with community foundations across the Lake Erie region. We're working with 7 communities to bring new technical tools to volunteer water quality monitoring groups across the region. One of these tools you'll be hearing about in depth today, the Erie Open Systems Spectrometer, a low-cost method for monitoring for nutrients that we're working with these groups to implement. We're also working to implement a common data infrastructure using the platform Water Reporter, developed by Chesapeake Commons. And this program, which we're calling right now Smart Citizen Science, is in its first of 3 years. So we're really excited about the potential for this project to scale beyond the initial partnerships we formed. You know, we're working very closely with 1 or 2 organizations in each of these communities, but we know that there's a really broad ecosystem of folks engaged in this space of volunteer water quality monitoring. I'm sure a lot of you on the call are engaged in this space. Many of you, we've probably already talked about it directly, but we're really excited to continue to work on this. And if this is a project you're interested in getting engaged with, if you're based in the Lake Erie region and you work with volunteers or are a volunteer to monitor water quality, we'd love to have conversations with you to figure out how we can better partner moving forward. I should mention also that we will be having a follow-up webinar to this session that will be focused on the other key technical tool I mentioned, Water Reporter. That webinar will take place on July 23rd, and we'll be sending the info out as a follow-up to this call. I have a third slide, right, where I'm introducing you, Kelly. I want to make sure I'm not doing it here. So it's really my pleasure today to introduce my colleague and today's key speaker here, Kelly Simon. Kelly is wrapping up now her postdoctoral degree at the University of Akron in Integrated Biosciences. She's been engaged with the Erie Open Systems team for a number of years, actually winning one of the prizes at our first Erie Hack competition in 2017. And now through this project and others, is actually working part-time with us at Cleveland Water Alliance as our senior scientist. So we've really been very fortunate to have her as a collaborator, staff member, and real leader in this space of citizen-collected water data. I'll be around throughout the presentation and around at the end for questions as well. but Kelly's really going to be kind of in the driver's seat for this session. So please, join me in welcoming Kelly to kick off the presentation now.
Thank you for that round of applause, everybody. And I just
Yes.
want to for coming and for voluntarily coming on to another Zoom call. I have tended to call myself a bit of a Zoom zombie, and so for voluntarily coming on and subjecting yourself to another one, I'll try to make it informative and entertaining. And from what I hear, we have a whole range of folks that are tuning in. So people that are kindergarten teachers on up to higher education professors, water quality professionals, to people that don't even know what a spectrometer is, but would like to learn. So I'm gonna try to cater to both of those spectra. I just made a spectrometer joke. And try to kind of keep everybody engaged. And I really had the kindergartners in mind, because the person was like, are we really gonna be able to teach spectrometry to kindergartners? Yes, and I will show you how. So I first wanna discuss HABs. And I think going into what we're going to talk about really is this little spectrophotometer. We really have to understand what harmful algal blooms are. And now this has a whole new meaning if you are from Toledo or know the context of this particular slide. If you don't, this is really a watershed moment, so to speak, in the Lake Erie watershed. In Toledo, Ohio, in August of 2014, Toledo's water was shut down to nearly 500,000 people. It made international news, all over the place. It was shut down for about 3 days. And really, it disproportionately affects everybody, but disproportionately marginalized communities, disabled communities, and if you were pregnant or a small child or ill, then you are immunocompromised, then the water was extremely toxic. And so what you wanted was obviously fresh water, but you didn't have it. So you couldn't just turn on the tap and wash your hands. And so we obviously don't want this to happen again, but why did it? Well, these harmful algal blooms come from a number of sources. And in the western basin of Lake Erie, where Toledo is, it's very shallow, it's very warm, and it's a very heavy agricultural area. So there's a lot of excessive nutrients that are run off. Now, we don't want to just blame all the farmers. And so there's other reasons as well, right? So there's urban runoff. There's excessive, the biggest part of agriculture is actually our yards. So you see all those really nice fertilized yards. And that is actually the #1 fertilized crop in America is our grass and our lawns. And so it's not just the heavy agricultural areas, but it does have a large contribution. And there's also a lot of adverse impacts as well. So our regional challenge here is that we will lose economically about 1.5 billion over 30 years if we don't actually find a mitigation or adaptation to these harmful algal blooms. Now, we do have a voluntary reduction of 40% by 2025. I would say that we're not really on board on that, but we're still doing all that we can to actually reduce it down to 2008 levels. But we don't really know. We have this insufficient data. Why do we have insufficient data? Well, it's very expensive to monitor. It's very time consuming. And so we just don't have the spatial or the temporal data sets to really understand those long-term trends in the watershed and where exactly those hot spots are in terms of nutrient loading. So we can't necessarily make like functional progress if we don't understand where those nutrient loading trends are. And we can't really be efficient with our investments if we also don't know where to invest into certain projects to mitigate the phosphates and nitrate runoffs. And therefore, we don't really know what our return on investment is when we actually do a mitigation project. And so we really need a lot more data. And so, let's see there. And then, so let's see what causes harmful algal blooms. Well, we've got, this is pretty much the watershed. We've got a lot of rainfall, it's agricultural runoff, and then you've got the residential runoff. That's your urban runoff. So whenever you see, a big heavy storm and the water runs off into the drain, that essentially goes into Lake Erie if you're in this watershed, or into a different watershed. Forest runoff, wastewater treatment plants that have, pipelines that go in. And then it goes into the water, and especially in the Western Basin where it's shallow, it warms up very quickly. And then you have this cyanobacteria bloom. And so I wanna, for the kindergartner teacher out there, in Finding Nemo, you had Mr. Ray, who was the teacher for Finding Nemo. He goes, quickly kids, gather, it's a stromatolitic cyanobacteria. And cyanobacteria in and of itself, this is how life started on Earth 3.5 million years ago. So it's not necessarily a bad thing, but in this context, it's very bad. And so it's essentially, it's not algae. What it is a photosynthesizing bacteria. So these photosynthesizing bacteria look like algae because they form these green, kind of thick, dense, goopy mats. And then it dies off, and then it creates a hypoxic zone, so these dead zones. And so there's a couple of different reasons, not just for why we have all of these harmful algal blooms in Lake Erie, but also around the world as well. It's a problem globally. One of them is in the Northeast Ohio context. All that really, really lush farmland out in Northwest Ohio, it used to be a swamp. So it used to be a wetland. This is a hardwood wetland. And what that does is it slowed down that hydrological flow. It allowed for the uptake of nutrients. And let's see. I don't know why it's not advancing. Hang on. There we go. So then we have this hardwood forest. Now this used to cover all of Northwest Ohio, the majority of Northwest Ohio, until we learned that, the settlers, I should say, learned that you can drain the swamp, and that's really, really fertile farmland. Now this used to be occupied by a bunch of Native American tribes. But Andrew Jackson in the 1860s said, you know what, let's move these guys off of this land because we're going to take it over because it's really fertile ground for agriculture. So instead of having this really nice, slow hydrological flow that entered into the Western Basin of Lake Erie, now what we got was we mechanized the reduction of the Buckeye, or I'm sorry, of the Great Black Swamp. So essentially, we sped up that hydrological flow. So we did not give nature enough time to uptake those nutrients. And in fact, now we have about 20,000 miles of drainage ditches, and that's specifically by design to move the water as quickly as possible. So the system is switched from a nice, slow hydrological flow with plenty of biomass to uptake those nutrients. But right now, it's designed to get rid of the water as quickly as possible. And that's because the farmland with the water table is quite high. So if you're in Northwest Ohio in particular, what you see are these called drainage tiles. And they're a very efficient way to move water off of the land. And if you look, here, this is a really nice graphic from Ohio State University. That water table, because it was a swamp, the water table is quite high. And so the yield, the plants won't grow very well. So they put those drainage tiles under the soil to move the water quickly off the land, bringing soil and nutrients with it in the process, and then extruding it out into the drains. Which some of them are so big they look like rivers in and of themselves, and then they join the watershed, and it quickly moves into the Western Basin. And so that's one of the reasons why it happens so quickly, is because it's designed to happen that quickly. And so, not to be too corny, but when I look at this, I see two things. I see like the absolute best sweet corn that you'll ever have, Ohio sweet corn, but it comes at a price because this is a wetland to me. And so when I go driving through Northwest Ohio, I see wetlands where you know, our 4th of July barbecue sweet corn comes from. So I think one of the main things I really want you to take away on this is viewing this a little bit differently, and understanding that the hydrological flow really contributes significantly to the harmful algal blooms. But you know, in addition to the spectrometers and citizen science, we have an awesome asset in Northeast Ohio, and that's the people. We have worked with so many amazing people that want to get involved. How do we help stop the harmful algal blooms? Here's an idea I have, and this is part of the project that we're doing with our spectrometer. And so we've got so many people involved in wanting to get out there and get in the watershed to help us build up those data sets so that we can understand both scientifically and then to actually inform our policymakers on how to change the regulations or understand or put them into a direction of where these nutrient hotspots are so we can do mitigation in particular areas for a higher return on investment. And so when we look at this, look at the data and the citizen science and with the professional data that we have for water quality monitoring throughout the Lake Erie watershed, you know, in that Northwest area, there's some really great data sets with Heidelberg. They've been going on since 1975. And in fact, it's the longest running data set in the world for water quality and nutrients. So like we know what's going on over there pretty well. We don't know necessarily some of the smaller tributaries as well as the broader watershed of what's happening. And so this was really the catalyst of, you know, for me wanting to do my own research. I wanted those data sets. I wanted to understand, you know, what were those long-term nutrient spatio-temporal trends for nutrient loading, and when did it flip the system to harmful algal blooms? And we know, but there are also some cross-scale feedbacks happening in that as well. And so I thought, well, if we don't have all the data, why don't we just make something to gather the data? And so, you know, a lot of times, here's the academic side. So there's a ton of spatio-temporal data points that we just need. And there's a fundamental mismatch at a lot of the scales as well. And so there is a gap that exists between citizen science data, high quality citizen science data and policy. And I'll say that that gap is getting a bit smaller. And especially at the local and the regional levels, the citizen science, there's some awesome citizen science and water quality work going on. It's often fragmented with protocols different geographically along the groups. And so one of the things that we're doing is we're harmonizing all of those protocols to get it to the regional scale so that we can analyze the data geographically from the entire region. And one of the things is the equipment is ridiculously expensive for a citizen science program. I mean, it's really expensive. This is a Hawk 3900 colorimeter. This is about a $3,500 spectrophotometer. And this is EPA approved, but it's a bit of a black box. You stick your sample in, you close the lid, put in the reagents, and then press a button and kind of beep boop out the numbers. Don't really know how it works. It's proprietary. And so, you know, the stuff for citizen science, it's lower cost, but it's not that accurate. This is on the very low end of cost and accuracy. It's public lab spectrophotometer made with cardboard and rubber band, as well as a Lego one, which you can totally do because it's all open source if you wanted to play around with it. But we wanted to blend these two so that we could fill that gap between citizen science and lab developed spectrophotometry, and to be able to get out there and get reliable data so that we could inform policymakers and watershed managers. So the main purpose of this was twofold. We wanted to bridge that accessibility and reliability gap, and then really figure out how can we make it the most robust, both in terms of protocols and the science, policy science, as well as in the laboratory science, analytical chemistry, and the optical physics that go into developing this spectrophotometer. And so the reason why this hasn't really been done before, even though there is a high need for it, is because, well, it was incredibly difficult. It was really difficult. And it would not have happened had it not been for the Erie Hack platform and Cleveland Water Alliance, because it really provided that platform and the motivation to get the most diverse interdisciplinary team together to actually tackle this one challenge. And so that's what we did. And it's taken us about 3 years to get to this point, and we're not done yet. So there's plenty of work to do. There's plenty of research to do as well. Okay, so what is a spectrometer? When I first started out, I'll be super candid, that word was scary to me. I did not like chemistry in high school. I avoided it. But this is why I went into biology, and then you partner with an analytical chemist. So I got smart on that end, but it was a scary term. And it shouldn't be, because this isn't a scary thing. All it is it's a way to measure a type of spectra or something, right? So you have a mass spectrometer, you can put something in it and beep boop out the numbers, and it'll show you exactly what that thing is made of, right? And what the spectrophotometer does, it measures a spectra. That's the difference between a spectrometer and a spectrophotometer, is that it measures a wavelength. And that's it, some type of light. So it could be UV, it could be infrared, near infrared. In our case, it's the visible spectrum. So what we do is we bring those invisible nutrients like phosphates and nitrates into the visible spectrum with those reagents. And then we shine the light through the spectrometer, through that light source, and there's a diffraction grating. And remember when you were a kid and you used to love playing with prisms? It's the same thing. So it's just a little, diffraction grating is like a prism. It splits the light, and then it filters it through the cuvettes that are there. The cuvettes are just containers. And there's a sample, so like something that you get from the watershed, and just some deionized water or diluted water. And so, and then it reads the different absorbance spectra from those wavelengths, and then we just calculate it. And I'm making it sound like it's super easy. It took 3 years to get to this point. But what we do is we have an app on the phone, and we created a Python code on the back end, and that's where probably a lot of the work came in. And that we read the different spectra of the sample and the control. And so when it comes up, we can get the absorbance value on your phone. And then we can upload it to Water Reporter, and then we can get that geographical pinpoint of the phosphate or the nitrate, whatever you're measuring. And so really it's just a way to split the light. And for my Gen Xers, or not Gen Xers, yeah, maybe Gen Xers, that's Pink Floyd. Pink Floyd had the prism on here. So it's not a scary thing. And then on the right, it's there's a lot of tools that you can use pedagogically in K through 12 that have really great things like the science of spectrophotometry. They don't say spectrometry, but that's what it is basically, splitting of light. And then for the science-minded, we've got the mathematical calculation over here, which is absorbance and log 10 of I over I naught, which is how we measure the intensity of the light. And so what we did was Erie Open Systems. We have come a long way from Erie Hack. Erie Hack started out as a proof of concept in an cut up Amazon cardboard box where we had to get the flap up so that it was aligned with the slit in the light. And so we have come a long way to this basically. And this is all open source. You can make one if you'd like. And this is just laser cut and 3D printed, resin 3D printed, not extrusion, but resin works best. And what you see here, you just put there's two cuvettes, one with a control, and then you have one with your sample, say from, Cuyahoga River that's in my backyard. And then let's say we're measuring phosphates. So phosphates, to bring it into the visible spectrum, it turns blue. And we stick that sample in there. We follow the protocol for the reagents. And then we just turn on the light in here, bring up the app on the phone. And what you see on this bottom left-hand side is the dual spectra that comes out. And then the app analyzes those two spectra to see what the absorbance is. And from that absorbance value, we can get the quantification of phosphates or nitrates in that sample. And so, again, the reason why this hasn't been done is because it is quite difficult. But what we found, this is our big finding that we're writing up for publication at the moment. We get a really nice log linear Beer-Lambert curve. That Beer-Lambert curve, even though it's a straight line, that's exactly what we want. And it just shows that it's exactly in calibration with the absorbance spectra. And this phosphate calibration curve is from the Erie Open System. And for the math and stats people out there, we got an R-squared R value of 0.97 on it. So we're really happy with this return. And what we found, though, our big finding was that you saw that $3,500 hot colorimeter. It doesn't matter what the optical hardware is. It does not matter. It could be this. It could be that $3,500 component. But what matters, the limiting variable in getting a really nice response on this Beer-Lambert curve is the chemistry. And so getting the right reagents, which we haven't necessarily found really fantastic reagents that work perfectly. The Hawk ones that are EPA approved, we are wholly not impressed. To say the least. So what we did was we used fish tank reagents. It's API fish care. And what we found is those give just as good of a response than those Hawk pillow reagents that are proprietary and very expensive. So that's what we've found. And we're really happy with that finding, because what that means is that we have the science behind it, and we can start to field test it to work with citizen science to get that data set where we need to be so that we can start to tackle those harmful algal blooms. So I want to introduce our team because we've done quite a bit, and like I said, it's really hard because we basically bootstrapped the entire thing. This is essentially a side project for all of us. We have won Erie Hack, third place in Erie Hack in 2017. On the left, we got first place in the University of Akron's Social Entrepreneurship Challenge. And we've gotten little mini grants all along the way to really just kind of keep going on the science in the lab. The upper right-hand photo is our original 5 teams. We have Dr. Zhencheng Feng, who's a mechanical engineer, Dr. Hunter King, who's our optical physicist, myself, who's a PhD in integrated biosciences and I do social ecological coastal resiliency, Dr. Adam Smith, who's our analytical chemist, and actually Dr., as of yesterday, Dr. Banafshe Kakipur is our computer scientist and data scientist. So we've got a pretty strong interdisciplinary team and that we've completely bootstrapped. On the left-hand side is our company logo. If you know a little bit of Chinese, that's the Chinese character integrate for shui, it's for water. So that's our logo. And on the right is we have a website as well where if you wanted to, you can go forth and you can build, you can build it, you can tinker with the code. Everything is open source. And I'll have the website afterwards. And we've done a lot of stuff. Now I'm going to put a little asterisk on here because this is, pre-COVID. But we want to make this as accessible as possible. So on the bottom left is when we beta tested it with first graders about 2.5 years ago. could first graders follow a protocol and understand what a spectrometer was? And I'll tell you, a lot of them didn't have that biases of that scary word of spectrometer. So they were fighting over using a spectrometer, using pipettes, using cuvettes, and they were way better on smartphone apps than a lot of the adults that we worked with in the maker fairs. So kids follow things precisely, and they were really accurate with measuring things to mix the reagents. So they did phenomenal. And then we've done a couple of maker fairs, 4 or 5 maker fairs, Wayne County, University of Akron Wayne, as well as the Akron Maker Fair and Cleveland Maker Fairs. So we've been all over doing a lot of outreach. And what we do is you can make your own spectrometers. So at these events, we not only teach about spectroscopy and harmful algal blooms, just to get people acquainted with, demystifying what this is and if they can get involved, but you can make it out of a cardboard tube. And then there's a couple of different ways you can do it. You can use the diffraction gratings that we get, which are super cheap, or you can use a CD. And along the way, you can also teach your kids what the heck a CD is, because half of them don't know what it is anymore. And so you can do that in your classroom. And so there's multiple ways that we're using this device for citizen science. So we're beta testing it out in the field this year. I have our collective arch nemesis on the right here because that's been slowed down a bit due to the coronavirus and COVID-19. And that's in both institutional and statewide regulations. So it's been significantly scaled back, but we still are going forward with each of these communities and local champion organizations. They all have these Erie Open Systems spectrometers that they're beta testing out in the field. And actually what we're doing is it's all driven to get towards a policy that can be, to drive policy. And that is this fundamental mismatch, this federal 2017 Crowdsourcing and Citizen Science Act. This was one of the last things signed by Obama before he left office. I believe it was in January of 2017 he signed it. And that was for all the federal organizations, federal agencies to institute citizen science protocols in their schematics. So, but the problem is, there's a huge gap between the trust and validity of citizen science, as well as, can you really trust a first grader to go out into the watershed? So there is a fundamental mismatch in terms of the trust of, real science and then citizen scientists. But for us, everybody's a scientist if they want to use one of these and go out into the watershed, because not only is it's easy to use, but it's fairly accurate as well. And so what we want to do, everything that we're doing with this GLOW program, is to get to that point where the policymakers and decision makers within this watershed will trust not only this data, but all the other citizen science data collected as well. So this is the science behind driving this whole program. So it's not like we're just going out and then it's going to go into a database in one place and then just sit there. What we're doing is we're taking all of that historical data that we have that's been collected throughout the years. We're taking all the new data that's going to be collected, not just on the EOS, but with the other citizen science tools as well. And we are instituting harmonious protocols throughout each of these communities so that they're all collecting the same things in the same protocol. And it's all going to Water Reporter so that we know exactly when it was collected. We know exactly where it was collected. We know who it was collected by. And ultimately, because we know who was collected by, we know the protocols that were used because they're trained. And so what we're doing is ultimately getting it to the point to trust that citizen science data. And in addition, there's a huge uptick in the citizen science academic literature, mainly because of the 2017 Obama's 2017 law. But really, it has just exploded in terms of both projects and the science behind it. And so we're going to also emulate this Albus et al. 2020 study, which really was the first citizen science watershed study to statistically analyze historically collected data and the professionally collected data. And what it showed was that as long as you have solid protocols across the watershed, and you have highly trained volunteers, then statistically, that data that a citizen scientist has collected is as accurate as a professionally collected sample. So this is a study that we'll be emulating throughout the GLOW project over the next few years. And then this is also really cool. We just got a grant through Microsoft AI for Earth, and we partnered with TechTown in Detroit, and Wayne State University has a big data and analytics group new master's program. And so we're gonna have students, master's students, use our data set to run AI and to really filter out through some of that noise that we're ultimately going to get through this work. This is, if we want to look at like what a neural network is, I love this photo. This came up on Dig. This is from an 1895 film to 4K. They used a neural network analysis to just crisp up that image. And this is what it does. So say we don't get necessarily perfect data all over the place, but we get enough data points, then we're going to be able to write that algorithm, train that AI, and be able to crisp up those signals that are coming through for phosphates and nitrates within the channels, within the watershed. So that's what we're doing. That's the next step. Although we're not going to have as much big data this year, just because our nemesis Corona, but that's okay. And we have a couple of papers. So if anybody is interested in reading the academic stuff, we do have one that's already published. It's the Journal of Chemical Education. We have a lot of pedagogy work that we do, both K through 12, but also in higher ed as well. And then the next one, we just submitted Hardware X, which is basically looking at picking it apart and having all the code processed. And so once that gets published, then we submit the Journal for Science of the Total Environment. And this is basically the culmination that shows basically what I just mentioned about the absorbance comparisons between both lab spectrometers. So it's like a $12,000, I call it the shoebox. It's like it's the size of a shoebox, but it's a $12,000 shoebox Genesis. And so we took that, and then we did a couple of citizen science lab kits as well as the EOS, and we analyzed all of that in the lab with all the different reagents that we could find. And so once that Hardware X is published, then we'll be publishing the third paper as well. But I want to bring it back to really what this is about, because the science is awesome, but we're doing it because what we care. This is a side project for all 5 of us, basically. We haven't gotten massive grants. We've been bootstrapping it. We work on weekends. We volunteer when we can, because it's a necessary tool for us to understand and to reverse these harmful algal blooms. And so our whole drive is to create the best blended product of the reliability that you can get from a lab developed spectrometer, and getting enough people out there that we can use, make it easy enough to use, and then get enough people out there so that we can get a ton of data points and just understand what those trends are. So there's multiple nested layers in terms of the analysis that we have going on. So it's not like we're just going to look at the data that we get back from a couple of images and say, okay, we know what's going on. So we've intentionally layered this and rolled this out very slowly, but very intentionally, so that we know the science is solid. And then we partner with not only Cleveland Waterlands, but some really great partners throughout the watershed that know about citizen science and volunteers way better than we do. And so because they're always out in the field, it's just an iterative process. They'll be giving us some feedback on what worked and what didn't and what they would like to see in this, and then we'll iterate it. And since it's open source, if anybody wants to go ahead and redesign it so it's a little bit more user-friendly, sweet. That's what it's there for, and that's why it's open. And we'll say our team is committed to open source. We developed this from public labs. So the idea came from public labs. And for us, public labs is all about open source and open science. And so for us, even in the journals that we publish, all open source. So we will never publish in, say, some of the closed journals if you can't access it. And so it's completely open. That's why it's gone a little bit slower than having investors come in and close that data and selling the data. But we're really proud of how far we've come on it. So, I can't say that there's a lot going on right now because, again, lower left, the coronavirus kind of put a damper on the volunteer season. And I know up in Michigan, they couldn't even get out for a while. But I will say, one of the really cool, unexpected, unintended consequences of this is that most of the citizen science groups that do all the water testing, they couldn't use their volunteers. So it was just the actual staff that were going out, but they couldn't use their lab because the buildings were closed. What the EOS has allowed them to do is basically set up a home lab. So they can still continue getting some sort of data coming out, even if it's not using their normal tools or in their lab. So if anything, we have at least some data that's coming out of COVID. So it's a tool that's bridged the gap in a number of different ways. And this one is an unexpected one in that most of the staff have their little home labs, and it could just be in your kitchen because, it's small and it's light. So yeah, we're really excited, and there's a lot of ways that you can get involved. You can nerd out with us, and you can check out the code, and you can check out the optical physics of it, and read the scientific papers. Or you can get with your class and teach them, the finding Nemo's stromatolitic cyanobacteria and all about cardboard tubes, spectrophotometers. We love it all. We do it all. And we just want to be out there and support citizen science programs going forward. And so we're really excited to be able to be sponsored by GLOW and just keep moving it forward for the next couple of years to see, we're going to keep pushing this through the lab. We're going to keep refining the reliability of it. And we're going to hopefully change some policies for the better so we don't shut down Toledo again. I love your applause, thank you. And Max or Savannah, if you have, I think we'll have some time to take some questions. So I'll have Max and Savannah curate that.
Yeah, absolutely. Thanks so much, Kelly, for your presentation. It's very interesting. We've been having questions come in via the chat and the Q&A for a little while. So I'll go ahead and lob some of these your way. The first question came from Larry Shear. Is there a difference between spectrometer and spectrophotometer? Since both terms were kind of used during the presentation, could you dive into that a little bit?
Yeah, I kind of use them interchangeably, and it really just is a very nuanced way. So technically, this is a spectrophotometer because it's measuring a wavelength, right? And so it's a type of light. So photo like light. A spectrometer can basically measure anything, like a mass spectrometer, put like a rock in there or something, so it'll measure the mass and the chemical makeup of it. So this, a spectrophotometer, is a type of spectrometer, and it measures wavelengths. And this one is in the visible spectrum, but it could be infrared, near UV. It could be any type of spectrum. And I will say, if we can bring, the other cool thing about this is that it doesn't have to be just phosphates and nitrates. If we can bring something into the visible spectrum, we can measure it. We just have to calibrate the code on the back end in the lab. But we're already working with the caving lab for iron and silica, because they go in caving for 4 days and they have to cart everything in, and they don't want to cart like a big heavy spectrometer. So, you know, we're working on silica and iron for measuring in caves. So as long as we can bring it into the visible spectrum on this spectrophotometry device, then we're good.
So that actually plugs into another question that came in through the chat, which is whether you've considered adding NH3 to the parameters measured so that you can look at animal or human waste runoff in addition to fertilizer waste runoff.
Yeah, we have a lot of interest for, you know, other things, like a ton of other things. Even chlorophyll is one. For now, we want to nail down solidly phosphates and nitrates. Nitrates we've got down. Phosphates, we're still not at the level, the lower end of the spectrum that we really want, because it just takes a tiny bit of phosphorus to really affect the system. And it's notoriously difficult to detect accurately at a low level. So even ours, we get a little bit of fluctuation in the low level. So what we do is we want to continue on phosphates and nitrates before we move on to anything else. So we know what we've done right, what we've done wrong. And we have not only the process, but we're confident in that we can move on from phosphates and nitrates. But we can do anything in the visible spectrum.
Great, thanks, Kelly. Another question that came in from an anonymous attendee in the Q&A. Have we looked into the requirements for federal approval, and I assume also approval at the state level would be relevant here, for new sensor technologies? What is needed in order for data to be used in decision-making in the context of the Clean Water Act or other legislation? And so in that way, how does this device compare with perhaps more established technology like the Hawk device that you mentioned? Yeah,
so it takes a lot of money and a lot of cutting through the bureaucratic red tape of getting through that process. I'd say one of the biggest problems is, well, we've gone through the system a number of different ways, but each time it kind of ties us in a knot. And so it's not a really easy, efficient process. And so what we've done is we've looked at the federal guidelines. We've talked to federal EPA. We've talked to the state EPA, Ohio EPA. And each state EPA has different requirements or can adopt different technologies, you know, per their own state guidelines to implement it into their own protocols. So we've looked at that as well. Or you can get certified as an individual to take measurements as well. So we're looking at all of these ways, and we're looking at the different configuration of what is the best approach for us to do this, given time and money, and the limited resources that a lot of citizen science programs have as well. So the other approach that I'm taking from the science is we are making this the most robust, we're putting the science behind it, and we are being able to, you know, we're putting it not only from the optical physics standpoint, we're looking at it from the analytical chemistry standpoint, and like that Albus 2020 paper that I mentioned, statistically analyzing that data historically and with professionals. And so we're also doing those protocols. So we're setting it up in the most robust scientific way at multiple layers within this. So from a legal standpoint, I'll say there's two different ways. From a legal standpoint, all we have to do is persuade a jury or a judge. You know, so if we go up, if the EPA is challenged, or if something with the water quality is challenged, and it goes through the court system, we have enough because we can look at the science. But if it's with policymakers and managers, then you know it's a bit more of a difficult process actually. So we actually are working with the, oh gosh, it's going to elude me. There's a group in Chicago that's really, it's a legal, an environmental legal group that's really interested in using this to monitor.
The Environmental Law and Policy Center.
I knew you would have it. Thanks, Max. Thanks for the assist. So we're working with them to monitor watersheds, and they can take people to court specifically if they're in violation of the Clean Water Act. And so, you know, from a court standpoint, and a legal standpoint, it's solid. But from a policy standpoint, it's a little bit different for us to really convince people to adopt the information. But we're working at it from a number of different angles.
And that's an area that we plan to continue to work on, or we hope to continue to work on over the next couple of years of the Smart Citizen Science project that we're engaged in with this device. Looking not just at how can this device be used in policy, but also how we can work to get citizen science, volunteer collected data taken more seriously, considered more credible from a policymaking and research perspective more generally, because that's also obviously a challenge, not just, how can new technology be considered credible, but how can volunteer collected data be considered credible? So those are a lot of big questions that we want to continue to work to answer. And for other folks, if there are other folks interested in those questions, we'd love to collaborate.
There's also a growing body of literature within that realm. So the growing body of academic literature, and if anybody wants to read up on it, it's really fascinating. There's a brand new academic journal, it's open source, that was started about 4 years ago now. It's called Citizen Science Theory and Policy. And it has some really great articles that deal with, they have a special issues just on policy. They have special issues just on water quality monitoring and citizen science programs, best practices. So there's a lot of really great information that's open source, open science, and freely available.
And some of that is actually referred to here in the chat. Anna, I believe Bogold, if I'm pronouncing that correctly, referenced a number of articles she's aware of about comparing kind of professionally collected data to citizen science data, as well as some conferences, including Society for Freshwater Science, that really have robust engagement in this space. So it is very much an exciting time to be kind of engaging with this question, or these questions. I think we have time for just one or two more questions here for you, Kelly. Jennifer asked in the Q&A, how are you working to QA/QC the data? Are you comparing it to grab samples, autonomous instrumentation, or remote sensing? How is that being approached as part of this project?
Well, let's see, we're not doing remote sensing on it just yet. For right now, it's looking at the professionally collected data side by side. And this really is our first year out in the field. And so we're starting with the side-by-side data. But I mean, ultimately what I would like to do is get that remote sensing, work with Joe Ortiz's group over in Kent State, because they do some really great hyperspectral analysis of harmful algal blooms. And it's not just about harmful algal blooms. They can actually detect the spectra of whatever different other kinds of organisms or algae are there as well. So from our data, we can actually flow it downstream and understand how it's impacting that Western basin through that hyperspectral analysis. That's down the road. We're concentrating more on getting this to a lower level of a detection so that specifically for phosphates. And learning how to scale up very intentionally so that we don't just kind of put it all out there and we can't control it necessarily, but starting very small and intentional and getting that feedback in an iterative process. And so that's what we're starting out with. But yeah, I agree. I would love to have some additional comparative data.
Fantastic. Yeah, and if there are folks, who might be interested in getting involved in sort of side-by-side testing, there may be opportunities for that. We're working to collaborate right now with kind of our core partners through the Smart Citizen Science program, but we are looking to evaluate opportunities for scalability in the coming years. Number of really interesting kind of observations here in the chat with the growth of home weather stations adding to the weather algorithm. It shows that home water monitoring kits could be next. That's kind of an interesting insight into the evolution of this space. Janina asks, she asks, how are we going to, but I guess maybe a good question to ask would be where can you go to see how the device works? And I would say right now, reach out to us by either email here. We have some resources that we've been developing over the past couple of months, a series of videos and protocols that, this is a good reminder for us, Kelly, that we should really be thinking about how we make these a little more public now that some more folks are more aware of the device. But there are resources that are accessible, and we're happy to share them with you if you're interested. So please feel free to reach out to us by either of our emails, as you can see on your screen, and we'd be happy to talk more. At this point, we're getting towards the end of the hour here. I'd like to invite you, Kelly, if you have any kind of final comments that you want to wrap up your presentation with, feel free to share them.
Well, yeah, I think, any advice, any suggestions, if anybody would like to use them or get involved in citizen science, there's ample opportunities, particularly if you're in Northeast Ohio. If you're a teacher, we obviously don't know what the next school year is going to look like, but we are working with the Leonard Gelfand STEM Center and working with higher ed and as well as K through 12. So we do have some educational resources. We do have some higher ed pedagogical resources. So there's all sorts of ways to get involved, whether you're a kindergartner and want to teach them what an ancient CD is and use that, repurpose it for their homemade spectrometer, or 3D print your own spec phone, which is what was in one of those first papers. So I can send you the printing stuff for a spec phone, which is a single beam. It's a little bit different, but it does teach spectroscopy in a different way. So just happy to get anybody involved, and it's all about collectively solving our harmful algal bloom problem so that we have fresh water for intergenerational equity.
Absolutely, really appreciate those thoughts, Kelly, and just want to echo that, for folks that are interested in this space, and particularly those of you in the Lake Erie region, we really are interested in figuring out how to partner with you over the next couple of years. I will say, it's been a whole process rolling out this project in the midst of COVID. So if we're ever slow to respond to you or anything like that, please do be patient with us. We're trying to make sure that we kind of get our boots on the ground with a couple of core partners, but are really in the process, we'll be in the process over the next few months of evaluating additional partnership opportunities. And our goal is really for this device to be part of a much broader network of community of practice around community-based monitoring, sharing tools, technologies, and best practices. And we want that to be as inclusive a space as possible. So with that, I'd like to thank Kelly again for a fantastic presentation. I'd like to thank all of you for taking the time today on this Tuesday afternoon to learn a little more about citizen science. And we'll be sending some follow-up information by email. But again, please feel free to reach out to either of us, if you have any questions or thoughts that you want to follow up with afterwards. With that, I'm going to go ahead and end the session today, and I hope you all have a good rest of your week.
Bye-bye.
Bye now.
Thank you.